And invasion of tumor cell via the AKT signaling pathway in osteosarcoma.
Dear Chairman, morning everybody. So our topic is to investigate the clinical significance and the biological activity of CEP55 in OS. So our methods include patients and tissue samples, immunohistochemical staining, immunofluorescence microscopy, cell culture, cell viability, cell cycle, apoptosis, migration and invasion essence, and the tumor growth, microarray processing, RT-PCR, and Western blot. So the results. So from the immunohistochemical staining, we can see that the CEP55 protein showed an obviously higher staining density in the OS tissues. We can see from the right picture that CEP55 was primarily located at the plasma membrane OS cells, and the staining is red. So the QRT-PCR showed significantly higher expression of CEP55 mRNA level in OS tissues. And also the Western blot confirmed the same result. And the overall survival rates of OS patients were associated with the overexpression of CEP55. So the tumor size, tumor metastasis, and 5-year survivals are associated with the overexpression of CEP55. So we can say after we knock down the CEP55 and the QRT-PCR Western blot markedly decreased CEP55 expression in the transfected cell lines. And also we can see that after knockdown of the CEP55, the cell had lower proliferation activities than the control study groups. So from another cell lines, we can see that after knockdown of the CEP55, the proliferation was inhibited. And we injected the OS cells transfected with the short hairpin CEP55 into the posterior flank of the known mice. And the results show that the tumor size was smaller in the study group than the control group at 28 days after inoculation. So from the facts analysis, we can see that the majority of the cells are arrested in the G1 phase, and the percentage of the cells decrease in the S and the G2 and the M phase. And from the flow cytometry, we can say that the percentage of the apoptosis of cells were highly detected in the study group. Also, the caspase 3 and 7 were highly detected in the study group. So after we knock down the CEP55, we can see the cell migration and invasion. We can see that from these cell lines, the migration and invasion rates of control cells were 2.1-fold and 2.4-fold higher than those of the controls groups. Also, in another cell lines, we can see the same results, that the migration or invasion rates of the control cell were 2.2-fold and 2.5-fold higher than the control groups. So from the microarray analysis, we can see that there are about 700 genes were found in the study, and the green dose represents the download regulated genes, and the red dose represents the up-regulated genes. So from the functional pathway enrichment analysis, we can see that the CEP55 may mediate OS development via the AKT signaling pathways, and the 6 genes were highly detected in the control groups, and just 2 of them, FN1 and the CCT1, were highly detected or highly expressed in the OS tissues. And the overexpression of CEP5 are associated with the FN1 and the CCTN1 expression. Maybe the FN1 and CCT1 are the 2 genes of the downstream pathways of the CEP55 AKT signaling pathways. So the conclusions, our study provides evidence that CEP55 overexpression in OS is correlated with the clinical outcome, and the CEP55 may be a key regulator of OS proliferation, apoptosis through the AKT pathway, and the CEP55 may be a putative therapeutic target. Thank you very much for your